Thank you, Carolyn.
Hi. Thanks for inviting us, and thanks to the truly startling number of people who are here listening to this today. So for the last almost 2 years, the Genomics program has been working towards IQMH and now CAP accreditation for our whole genome and transcriptome assays. So I'm just going to take a few minutes and walk you through the accreditation process, and then Dax is going to give some more information on the actual validation experiments and our sample requisitioning system that's just been developed. And before I start, I'd just like to say thank you to Tissue Portal, who's been working with us very closely through every step of this process. Especially Ilinka Lungu has been really, really helpful and done quite a bit of work. We're not going to talk about the Tissue Portal process or portion of this today, just because I don't represent them, but just know they've done a lot of work towards this. Okay, my slides don't seem to be advancing. There we go. Okay, so what is accreditation? It's just official confirmation from a qualified organization that we meet some desired set of requirements. So this is a standard. Typically, it comes from ISO. ISO, of course, stands for the International Organization for Standardization. And I'm super dyslexic, so this acronym is just fine. This is the organization that develops and publishes international standards. So they cover absolutely everything from food safety to aerospace engineering. A standard is just a set of rules telling you how to operate within some kind of industry, usually developed by a team of experts. And the standards are just audit requirements. So ISO doesn't come in and accredit you, they don't audit you, they leave it to another organization to do that. But the standards that are applicable to us here are 9001, which refers to quality management systems, and in particular 15189, which is quality management systems in medical laboratories. So the two organizations that are going to accredit us are the Institute for Quality Management in Healthcare, or IQMH. This is a Canadian organization that will audit you directly against ISO 15189 requirements. Like they'll come in with the standard as a checklist and check you for compliance with that. So if you're going to do any kind of diagnostic testing in Ontario, you need to have this. And this was also formerly the Ontario Laboratory Accreditation. We're also applying to CAP. This is an American organization. They are very similar to IQMH, although they're a little less stringent. They accredit loosely based on ISO 15189. And the reason why we're going for 2 accreditations is that CAP, well, IQMH is probably better for us as an Ontario-based organization. CAP is just better known. And I'll get to if this is kind of related to my point 3 here, but why are we bothering to accredit the laboratory when it takes so much time and resources to actually do? So first of all, I think it shows an extreme dedication to quality. It's a goal for the team to show to ourselves and to our collaborators that we are a high quality organization, that you have to monitor your lab environment, all your processes, document your training. It's a lot of work, and it's evidence that you care about quality. And also the timing was right. We had experts already present in the program, and there's a lot of expertise now. So we've got 2 auditors, the quality manager, and Trevor Pugh joined our program. Pardon me. Trevor Pugh joined us as medical director, and he's certified by the American College of Medical Geneticists. So this now allows us as a program to release clinical reports. And then thirdly, clinical trials have all started requesting this in some form. They might not always know what kind of accreditation they want, but they want something, usually CAP. So by bothering to accredit our lab, this greatly increases our scope and the amount of work that we can actually participate in. In this slide here, I just put this up to show that quality affects every department in the program, and our program at OICR is the merger of 3 different departments. So there's genome sequence informatics that maintain, develop the informatics system, the pipelines, translational genomics laboratory that is focused on clinical reporting, the genome research platform that operationalizes all the assays and then is focused on the research use only work. And then uniting all these departments is the quality assurance oversight. So this focuses on all of our SOPs, all of our shared space, common protocols, pipelines, reporting processes, laboratory information management system, equipment monitoring, and project lifecycle. And this is an example of the application cycle for CAP. IQMH is really similar. So normally a lab would have to have been working for about 2 years to get themselves established, just to be ready to even apply. And then you have to participate in a proficiency testing program for 6 months. Then you can request your application. They give you some specific checklists. You schedule your inspection. They come and inspect you, and then the auditors will notice any non-conformances you may have. You have a month to resolve those, then you're awarded accreditation. Afterwards, you get audited every 2 years to make sure that you're still compliant. And right now, we are at the step where we have, we're just scheduling our audit probably for December. But on this slide, I want to show you just how much work we've actually done from this point, starting in May 2018. The first step is always to create the quality management system, and we had to build ours from nothing. So that involved creating all of the quality, safety, technical SOPs, and a document control system to maintain all of those SOPs and other related documents. We set up a lab monitoring system, so that's fridges and freezers, internal temperature, humidity, etc. We have a CAPA system, so that's corrective and preventive actions. What this means is that for all your SOPs, if you have a non-conformance or a deviation from the protocol, you have to document this, and you have to have a system in place to correct it at the time and prevent it from ever happening again. So we have that in place. We have all of our equipment on maintenance schedule, and there's a lot of documentation that goes along with this. The same goes for our inventory. We built an inventory management system called Ramen to go with our LIMS MISO. We worked with procurement to set up regular vendor evaluations, because when you lock in assays, you also lock in reagents. So any vendor providing those to you becomes critical. And we track all of these key performance indicators. So, I mean, that could be anything from turnaround time to number of safety incidents reported to just number of assays completed. Those are all tracked regularly, and they're reviewed annually at our management review, which is the most important meeting that you have annually as part of the quality management system. All your department leaders get together, review every aspect of the system, and make sure that it's fully functional. And then we're finally just wrapping up the assay validation that Dax is going to describe. After that's fully finished, all of our personnel have to be trained in the assays that we have just validated and fully documented, at which point I'm going to conduct an internal audit with the audit team here that will prepare us for our 2 external audits that are going to come from CAP and IQMH. And then hopefully around March 2021, we should be fully accredited by both organizations. So I'm going to stop sharing my screen here now and pass over to Dax.
Okay, everyone can see the title page there. Thanks, Carolyn. So we're going to offer a very standard suite of assay items, including whole genome, 80x tumor, 40x normal minimum coverage, and tumor transcriptome, along with targeted ctDNA panels. Our intake will include tumor FFP tissue section, so 10 times 10 micron slides for tumors that are the size of a pinky nail. Sorry, I'm getting a prompt to unmute myself. Okay, so for larger tumors, it'll be 5 sections.
We can't see your presentation. We can only see your PowerPoint.
Oh.
Window.
Okay, sorry.
I think you need to go to presentation mode because I can see the outline.
I'm also happy to present on your behalf, and you tell me to change slides.
Okay, sorry. Must be this one. Did that fix the issue?
No. Dax, I will share your slides.
Okay.
Okay, go ahead.
Sorry about that. So our service offerings will include minimum 80x tumor, 40x normal, and tumor transcriptome, along with targeted ctDNA panels. For tumor FFP sections, for larger sized tumors, we're looking at 5 slides at 10 micron. For smaller tumors, about pinky nail size, we're looking at 10 slides, 10 micron each, with pathology review on the H&E. For fresh frozen tissues, we do take in both fresh tissue and LCM material, matched Buffy coats, and for upcoming serial liquid biopsy and ctDNA-based panels, vials of Strep, 3 vials of Strep tubes or assay time point. So our workflow begins with the requisition process. Upon receipt of a requisition, samples are shipped through to tissue portal, allowing for accessioning and extraction. Then materials are transferred to the Translational Genomics Lab, where licensed medical laboratory technologists synthesize the library. Materials then pass over to the Genome Research Platform for sequencing on NOVA and NextSeq Illumina platforms. After a data analysis, it passes to the Cancer Genome Interpreter team for processing and annotation. Finally, we move on to sign out by University Health Network clinical medical geneticists. Next slide. So the advantages of these accredited services is complete profiling and de novo discovery opportunities. Our service platform requires both whole genome and transcriptome tumor profiling, which allows us to integrate these data sets together in our analysis to help verify things such as amplifications or fusion events that we might see breakpoints for, as well as a variety of other informatic analysis. For serial ctDNA options, this allows us to look at minimally residual disease, potentially very early detection, and minimally invasive and clinic-friendly screening programs. All of the work, again, is completed by medical laboratory technologists and certified clinical medical geneticists or Board of Pathology certified molecular genetic pathologists. These clinical reports that we will generate can be entered into electronic patient records and can allow for the appointment of patients to local clinical trials or to alter their treatment therapies based on molecular resolution that we may find in their genomic profiling. As well, the data that we generate from whole genome and transcriptome can be used for other research-only initiatives such as ICGC Argo, or as long as we have consent processes in place for the individual clinical study. Next slide, please. So everything begins with an online requisition. So we have created an online portal system for management of requisitions. After your legal agreements and ethics approvals are in place, we will launch your study approval. We will set up accounts for your clinical coordinators. Clinical coordinators will be able to recall their requisitions. They'll be able to submit formal consent withdrawals, and they'll be able to download their submissions for each study that they have access to. We collect minimal data elements, including things such as study and study ID, study sample ID, site of biopsy, cancer diagnosis, and we use standardized OncoTree codes for tracking tumor information. This system has role-based access, so there are dashboards for requisitioners versus medical geneticists, and you can also download the clinical report if your role permits that utilization. Thank you. Next. So our report is going to give you general streamlined metrics, including mean coverage, callability, purity, ploidy, tumor mutation burden, and signatures like HRD and MSI, fraction of the genome altered, single nucleotide variants, indels, copy number, expression, and fusions. Next slide, please. So we are aggregating a significant amount of information because the platform is based on whole genome and whole transcriptome. We will only be reporting out evidence levels 2 and greater, though we will be annotating all results using standard OncoTree codex. Next slide, please. So we've undergone an extensive validation of all of our processes, and that starts with tissue. So we've looked at DNA and RNA co-isolation from FFPE tissues. We've looked at fresh frozen DNA and RNA extraction, either macrodissection or laser capture microdissection, buffy coat extractions, and plasma isolation and ctDNA extraction. For library synthesis, we've used kappa hyperprep for whole genome and kappa hyperprep for targeted ctDNA panels, as well as Illumina TrueSeq total RNA library preparation methods. We use a variety of automated processing methods, and so as part of our validation, we have looked at our whole genome analysis pipeline, as well as Transcriptome and our targeted sequencing-based UMI panel processing. Next slide, please. So there are four main pillars of laboratory assays, which you can see here, which include nucleic acid extractions. We take in your sample tissues and blood products, and then we build our whole genome, whole transcriptome, and targeted ctDNA libraries within the laboratory space. Next slide, please. We have an integrated system that tracks all of our samples as they are received and moves through our different workflows. Using our library information system, we queue up all libraries onto the sequencer, which allows for immediate detection after run completion and an immediate launch of all associated workflows, whatever that assay may be, for review by the Clinical Genomics Interpreter team. Next slide, please. So one of the parts of our validation is to use real-world tissues. So we've, as everyone has probably experienced, we've had hiccups with some commercial controls. So we have used tissues that have been provided by the Ontario-wide Targeted Nucleic Acid Study, as well as samples collated at OICR through the Ontario Tumor Bank. We have used tissues from the PanCureX initiative at OICR, known as COMPASS, and we have also used a variety of buffy coat and plasma samples. For our whole genome validation, we have looked at tissues from FFP, fresh frozen, and LCM. We take 3 stages to our validation, the first being analytical sensitivity, evaluating what are the lower limits of our assay where we can still receive functional information from, and settled on 25 nanograms for fresh frozen tissues and about 100 nanograms for FFPE. We've looked at the precision of our assays, which includes batch effects, operator variants, technicals, sample and sequencing replicates, and finally expanded out to clinical sensitivity. So we've looked at over 60 tumor normal pairs. 51 of these are FFPE using tumor tissues from both the Octane and React studies, as well as 9 fresh frozen tumor normal pairs from the COMPASS study. All of these samples have been externally reported at UHN's Advanced Molecular Diagnostic Laboratory. So we have reported variants that we are comparing our results back to. We look at copy number variants, indels, single nucleotide variants, tumor mutation burden, purity, ploidy, and a variety of other genomic events. For whole transcriptome, we also completed a similar series of analytical sensitivity and precision experiments and a clinical sensitivity cohort encompassing approximately 50 or so samples, looking for percentile and expression outliers and fusion. As well as these ongoing validation experiments, we've also participated in 2 rounds of solid tumor proficiency tests run through CAP. Next slide. So we have run into some challenges. It's a big scale up to go to whole genome and transcriptome. So as I mentioned, we have validated data sets that have been reported in clinical labs, but these reports in the past contain comprehensive gene sets with minimal footprint. So when we scale up from assays such as Oncomine to the 5' gene panel to whole genome and whole transcriptome, you can see we're moving from about 0.4 megabytes of target space up to 3 gigabases of expressed genome. So we run into that issue when we're looking at sensitivity and specificity of trying to scale our results to a very limited number of known targets. We also, because we've incorporated tumor normal sequencing, we have a little bit of a distinct advantage over some of the prior annotated data sets in that we can filter a little bit cleaner than some of the prior reported assays. But you can see in the variant plots below in the green little tick marks there, that between the two assays of whole genome and targeted panels, we have nice correlation, and we also see a lot of the mutations common in both the targeted panel and the whole genome. Next slide, please. So again, kind of carrying through that blowout into a whole genome sort of analysis, do we capture the things that we anticipated? And yes, we do. Here you're looking at CAP proficiency test results. We have 3 different samples that were sent to us, and we performed whole genome analysis. These are tumor-only sample sets, and we do find the variants that are anticipated by CAP, but occasionally we do pick up other things that we don't see on the clinical report, and that's marked by the red question mark there. And that draws into question, as you go into these validations and different cores within North America start to process whole genome data, these variant lists keep on expanding as greater and greater discoveries are made by integrating whole genome data. Okay, next slide. So where are we moving to? So as mentioned by Carolyn earlier, we're moving towards accreditation. So we need to start aggregating data from our clinical studies and reporting out our results. So we have a few founder projects that we're beginning with. So one of them is the CHARM study, or hereditary cancer study. We have the Ontario-based study Octane, which will also contain whole genome, whole transcriptome reporting. We have a gynecological study with UHN called Heartbeat OV. And finally, PASO-1, a pancreatic ductal adenocarcinoma study that will be launching soon. And all of these studies start to stream data back to medical reports in anticipation of the launch of the Marathon of Hope series of studies, which will form the vast majority of our research platform in the coming months. So at this time, I'd like to wrap up and open the floor to questions.